Boa tarde. Vamos dar início ao Fórum de Interdisciplinaridade do mês de abril de 2016. E nós temos hoje, então, a presença do Kevin Blighe. Kevin foi fundador de uma companhia de bioinformática clínica em Londres, onde ele ainda continua ligado, e ele é também um bioinformático na Queen Mary University de Londres, e diretor da Biotech Cell, e também bioestatístico na Biomarkers. He's going to speak us in English, so I'm turning now into English. Okay. His overall experience spreads across the Republic of Ireland, England, Italy, Malta, Brazil, and the United States of America. He has training in both the computer and biological sciences, thus allowing him to connect the information technology and wet laboratory worlds. I can see here in the audience some bioinformatics as well. We were expecting this meeting to be a meeting point for you, so I think this is a very good opportunity for all of us. And we thank you, Kevin, for your disposition to be here and talk to us about your... We discussed that perhaps we would like you to tell us about your personal experience, but of course that you can expand your speeches as you wish. With Kevin was very interesting because I found it very interesting, or I realized it's very interesting because perhaps it's already a commonplace to have people living, for example, as his example. He lives in Porto Alegre, has married. Oh, it's a difficult name. Please say it here in the microphone. She's a Brazilian.
Salwa.
Are you a nutritionist? Okay. And so Kevin is living in Porto Alegre. And I think he has at least one good reason to live in Porto Alegre. But he works in London or in several other places in the world. This is the new world. And we would like... yes, so... And I think it will be very nice to hear Kevin, okay?
So, thanks very much, Professor Teixeira, for the introduction. Like Professor Teixeira said, I live in Porto Alegre, but I still work with, I have my company based in London. I also work for Queen Mary University in London. And involves obviously a lot of travel from Brazil to London, which is costly, and it's a lot of travel on an airplane, but ultimately it's a good quality of life because Porto Alegre is a really beautiful city. There's a lot of trees here, and I think it has lots to offer. So I'm enjoying my time in Porto Alegre so far. I'm coming up to around 1 year living here, I think, already. So it's really good. So I'll start the presentation. This should be about 40 to 50 minutes long. And the title of the presentation is, if I can get this right in Portuguese, Construção, carreira, Bioinformática, experiência, pessoal. So tonight we're going to talk about bioinformatics. So there are three main sections to this presentation. So first I go over an introduction, and I talk about what is bioinformatics to explain it. Then there's a personal introduction of myself. Then I talk briefly about the future of bioinformatics. In part B, I go over practical applications of bioinformatics because bioinformatics would be entirely useless if it was not applied to, say, biological data and medical data in order to explain, for example, certain diseases. So in the practical applications of bioinformatics, I cover things like cancer, clinical diagnostics or genetic testing, B-cell lymphoma, and then rheumatoid arthritis. And then in the final section, I go over a kind of a an issue, as I've seen in bioinformatics and in research in recent years. I've entitled this section "The Complexity of Life" or "The Frontier of Bioinformatics." So, without further ado, I will start. So, introduction. So, what is bioinformatics? So, on a very fundamental level, bioinformatics is the bridge, a ponte entre informatica e biologia. So, bioinformatics is the bridge between computer science on the left and biology on the right, on a very fundamental level. And if you look at the word biology, and you look at, so biology is bio plus logi, which in Latin means the study of life. So if you look at bioinformatics, then the word bioinformatics, it's bio plus informatics, which is the informatics of life. So what is informatics? Well, informatics covers a lot of different kind of subtopics. So it includes computer science, information technology, algorithms, and mathematics. So bioinformatics is about storing and analyzing biological data. It's very broad or interdisciplinary, and it's an ever-widening area. And so other terms that you will hear in relation to bioinformatics include systems biology, computational biology, health informatics or clinical bioinformatics, and more on that later. And then another modern term which has sprung up recently is biomedical data science, and more on that later as well. So actually, well, not all of us, but the majority of us are already bioinformaticians. So on a simple level, if you use Microsoft Excel to analyze biological data, that's bioinformatics on a very fundamental level. On a more complex level, which is where I am, it's about writing new and creative computer programs and working with large data. And we do this to make the biologist's life easier. So this is just an example of a computer program that I wrote a few years ago. This is simple for me, but this is what I kind of do on a daily basis. I design programs like this. This is called a script. And as I say, a skilled bioinformatician writes these every day to just process some biological data and to interpret that data. And then pretty much we send the results back to the person who gave us that data. So this is just, this is actually called shell scripting. And this big dark screen is, it's a screenshot captured at the tail from a Unix operating system. So as a bioinformatician, I use this type of operating system every day. And a Unix operating system is very different from Windows, Microsoft Windows, or Apple Mac OS. So Apple Mac OS and Microsoft Windows are tailored for general use, whereas Unix is more tailored for, well, for bioinformatics and for computational methodologies. But you have to have a certain level of advanced knowledge in order to use this type of operating system. So I mentioned biomedical data science earlier, and this is just a new topic that has sprung up recently. So what is biomedical data science? Well, biomedical data science is the study of data derived from a medical setting. It's the structure of the data, how best to number crunch the data, and what the data means and shows. And to highlight the importance of biomedical data science at the moment, Stanford University in California have just created an entire department dedicated to biomedical data science. Another company in San Francisco, Genentech, or Roche, they're the same company, they have an entire department devoted to bioinformatics. We're talking about entire departments here, which is a lot of funding of money. And then so integrating biomedical data with patient medical records can make more precise the ancient art of medicine, I believe. And then as we keep generating more and more data, I believe we get ever closer to actually modeling life itself. And in doing that, we model disease as well, and we can perhaps help to explain diseases better. So I also mentioned clinical bioinformatics earlier, and my company based in London the focus of my company is clinical bioinformatics. But there's a big distinction between clinical bioinformatics and research bioinformatics. So what is the distinction? So in general, bioinformatics is mainly about the application of computational and statistical techniques to biological data, but clinical bioinformatics is then, it's particularly related to the application of these to biological data deriving from clinical environments, so like hospital environments, for example. And such data, so clinical data, typically requires a different analysis approach. For example, it can be of a highly sensitive nature and may even be governed or protected by law. And it also requires a different level of interpretation, because when you're analyzing clinical data, that's data from a patient who is probably waiting on a diagnosis. So the impact of the results can be greater. So there's the point I'm trying to make with this slide is that there's a big difference between clinical bioinformatics and research bioinformatics. So in bioinformatics currently, there's actually a great paradox. So currently, those who lead bioinformatics or head bioinformatic units, like myself, for example, we actually have no formal training in bioinformatics. So I never studied a master's degree in bioinformatics as such. I never did a bachelor's degree in bioinformatics. So we all arrive into bioinformatics indirectly via other disciplines. So in bioinformatics currently, you will see people with backgrounds in computer science, like me, electrical engineering even, mathematics, statistics, physics, and chemistry. And question, well, what do you study? If you study one of these disciplines, you have the possibility of branching into bioinformatics, of becoming a bioinformatician. And so, as I just mentioned, so these disciplines are now either working with bioinformaticians or they're becoming bioinformaticians themselves. But also there's another group of people that we need to think about. And that includes doctors, surgeons, biologists, and immunologists. So even these people are working heavily with bioinformaticians. And in my career already, I've worked with a few surgeons already who had an interest in learning bioinformatic techniques. They're surgeons, but they still had an interest. And obviously, in order to be a bioinformatician, you need data, and data is produced by technology. So on the left here, this is just a microarray chip. So microarray technology has been around for a long time. And it mainly analyzes gene expression data. On the right here, this is a modern-day DNA sequencer. This is an instrument by Illumina. Illumina is a company, one of the biggest companies in the world. So this is a DNA sequencer. It sequences DNA. It's a next-generation sequencer. And so I say at the bottom here that the best bioinformaticians can speak the language of all of these. So if you want to be a really great bioinformatician, you need to interact with all of these different people. And so you need to be able to talk to computer scientists, statisticians, doctors, surgeons, immunologists, etc. The more general knowledge you have from each of these disciplines, the better the bioinformatician you will be. And so I'll just go back. So once we get data from an instrument, the data is typically messy or dirty as such. It requires a lot of quality control, so we have to process the data, ensure that the data is of high quality and that it can be used to infer results, good results. So as I say at the top, a large amount of a bioinformatician's time is spent on quality control of data. So if we look at this image here, this is an image of this thing here. So this is just a microarray chip. So if you look at this, it probably means nothing at the moment, but each kind of point of light, each bright spot, is a point on the chip where a DNA molecule has bound to the chip. And when it binds to the chip, it then gives off light. It fluoresces light. And we can then infer gene expression, the level of gene expression, based on the intensity of the fluorescence. So as I say, this is a highly dense microarray showing fluorescent intensities. So what can we do with data that we get from this? So data that we get from this in its raw state will require a lot of quality control. And I'll show you what you can do with that. So when we get data from, for example, a microarray chip, if you look at it on its raw state, so this is a box plot. It shows the distribution of values from each microarray chip. So if you look at the black bar in the center, that's the mean value of the mean fluorescent intensity value from each chip. So as you can see, the mean is not level, it goes up and down. So we have to apply a normalization method to this in order to ensure that the mean is level across. So this is raw data, this is normalized data. So you can see it looks more tidy. And then from that, again, we can do further things. So one thing we can do is we can look at more box plots, and we can, for example, check expression of a gene between two different groups. So for example, if this is gene X, we can check its expression in group 1 versus group 2. And you can see its expression in group 2 its trend is higher, but its mean is actually slightly lower. And in these other plots, it's a similar story. And this plot on the right is a dispersion plot, which again is used as quality control. And these two plots, so volcano plot and then MA plot, these are widely used in microarray analysis in bioinformatics. And what these are showing are the results of a statistical analysis between two groups. So say, for example, if you have gene expression data and you want to compare healthy versus disease, so say, for example, control samples versus cancer samples, then you can check the distribution of p-values and log fold change values to produce these nice images. And again, these are just about sort of quality control of the data. And this nice image is clustering with heat map. So it'll take some time to explain this, but so what this shows is basically across the top you have a tree structure, and each vertical line is a sample. So then you have samples grouped into different groups. So you have a green, which is NPS. Then you have pink samples, blue samples, black samples, and red samples. And you can see that the green, pink, and blue samples are in their kind of own cluster here in the tree structure. I don't know if you can see that. Whereas the black and red samples are in their own structure here. So they're very different. And then on the left here, you have each horizontal line here represents a gene. And if you want to tell the expression of each gene in each group of samples, you have to look across the color here. So in this plot, if we look at the key, green is high expression, red is low expression, and then black is normal expression. So if we look, say, for this block of genes, this block of genes is lowly expressed in this group, so this block here. Whereas in the black and red samples, they're highly expressed because they're mostly green. I don't know if you can see that. This is commonly used in bioinformatics analysis. And so this image as well, this is a correlation heat map. It's a very pretty image, and what it shows is that it shows different groups of genes that are in high correlation with each other, so high or low correlation with each other. So this group of genes is in high correlation because it's a nice blue block. So this is the correlation scale. -1 is negative correlation, +1 is positive correlation. And this group of genes as well is in high positive correlation with each other. So now we move on to who am I? Well, I'm a computer scientist with an interest in answering biological questions. That's why I became a bioinformatician. And like Professor Teixeira said, I'm also a bioinformatic consultant in my spare time with my company. And so my primary skills are in next generation sequencing analysis, DNA and gene expression microarray analysis, biostatistics and algorithm development, software development from my computer science days, and then computer programming as well. And so, like everyone else who's now leading a role in bioinformatics, I also arrived into bioinformatics unconventionally. So, like I said, I studied computer science originally in Dublin in Ireland, where I'm from. Then I studied biosciences and industrial biology in Carlow in Ireland. And then I did my doctoral degree in breast cancer studies in Leicester in England, and my thesis was entitled Molecular Analysis of Circulating Cell-Free DNA in Breast Cancer Patients. So my background originally was in computer science, and then I arrived into bioinformatics due to my background in computer science. And so since then I've worked in quite a few different places. So in cancer studies in Leicester, in systems and computational biology in New York, in America, clinical diagnostics in Sheffield, in England, hemostatic disorders in Milan, in Italy, cancer studies at Imperial College in London, and then currently where I am at the moment is I'm working in rheumatology in Queen Mary University of London. And so, just briefly to explain the idea of what we do in computer science, and so this may help to explain why I then branched into bioinformatics. So in computer science, we develop software. We're trained to be software developers. So this is just a list of different programs that I've developed in my career so far. These are computer languages on the left, which some of you, the computer scientists among you, will know quite well. And I guess the interesting thing for biologists and doctors is that how can we translate this type of knowledge into the medical setting? And that's where bioinformatics comes in. So due to my knowledge of software development, I found it easy to transition into bioinformatics. So just a short slide on the future of bioinformatics. So 3 years ago, I predicted the following at a presentation in England. It was Manchester in England. I predicted firstly that bioinformatics will continue to expand and branch into other areas. So that's definitely true. It has certainly expanded hugely. I predicted that new university courses in bioinformatics will appear, and they currently are appearing mainly as master's programs in bioinformatics. So Master of Science in Bioinformatics, that's true. And then I also predicted that bioinformatic hubs will be built, which they are currently being built. So a hub would be a centralized, say for example, at the university here, a hub will be a group of people working together with the interest in being bioinformaticians and analyzing data, and they take data from different departments or faculties across the university, and they analyze that data, and then they send the results back to the relevant principal investigator. So that's just like a group of people who may be part-time in bioinformatics. What other universities are actually doing is they may actually hire new people, they bring in new people, give them a full-time salary, and they're sold that then the sole job of that person is to work in the bioinformatic hub to process data. So I guess it depends on the resources of the university. Or you get situations like in Stanford University where they've just created an entire new department devoted to bioinformatics and biomedical data science, but obviously they have a lot of money in Stanford in California. So does that answer your question? Yeah. Okay. So the key points from this first section then, at a fundamental level, bioinformatics is the bridge between information technology and biology. Bioinformatics is very broad, it's extremely broad, in fact. And I guess this is a key point for all of you here. You do not need to study a course in bioinformatics in order to be a bioinformatician. So like you saw with my background, I'm mainly a computer scientist, but I'm now a bioinformatician. And it's actually funny on this point because there was a time after I finished my doctoral degree where I was wondering, what am I going to do with my career? I was going down the route of working in a wet laboratory, so doing PCR, extracting DNA, and doing gel electrophoresis. But one day I actually decided that I'm going to start calling myself a bioinformatician. And when I started calling myself a bioinformatician, employers listened to me more, and I became more employable. And I did that mainly so that I would not lose everything that I learned in computer science. If I was going down the purely biological route, I would have lost all of my knowledge in computer science. So making that decision to call myself a bioinformatician professionally, it's changed my career and it's given me a lot of job opportunities. But that is a key point. You do not need to study a course in bioinformatics in order to be a bioinformatician. And then, so another key point, research bioinformatics is very different from clinical bioinformatics. That's definitely true. So currently, there's currently too much data to analyze in the world. As you saw, I have 4 different positions. That's because there's so much data to analyze. People contact me every day looking to analyze data. So there's currently too much data to analyze and too few bioinformaticians. So we need more bioinformaticians actually to process all of this data. And again, bioinformatics, it looks like it will continue to grow further in the future. So that's the first section done. Any more questions or?
I'm thinking here, when you try to find out something about statistics analysis,
yeah.
the major part of this effort is based on R language program.
Yeah.
And today we are trying to do this in Python and other languages.
You must use the microphone because of
Sorry for that. My question is based on an article I was reading today. The top 10 startups in Bay Area are based on big data and data science at this moment. So do you think that we moving forward using R like the main program language, or can jump to Python or any other easily?
Yeah, that's a good question. So OR is he mentioned OR. OR is a programming language that we use a lot in bioinformatics. So like I mentioned, programming languages on the previous slide, like Visual Basic, C++, Java, they are programming languages. OR, it's literally the letter OR, is a kind of, it's been around since the 1970s, but it's only now becoming more popular. in bioinformatics. So I use OR every single day of my life to analyze data. And it is the main programming language that I use to analyze bioinformatics data. And as it becomes more popular, well, so for example, to analyze microarray data, previously you could not use OR, you had to use other programs. Nowadays you can analyze microarray data in OR. There's a lot of different OR packages to do that. Regarding Python and Perl and these other scripting languages, I would use those mainly, if I cannot use OR, then I would use a different language. So shell scripting in a Unix environment, or Perl, Python, or C++. But OR is definitely the easiest to use because, yeah. Okay. The next section then is practical applications of bioinformatics. So here I go over, this is probably the most interesting part of the presentation because here I actually go over the different medical areas in which I've applied my skills of bioinformatics. So the first is cancer, which a lot of people have an interest in cancer. So let's just first look at how cancer progresses, which I'm sure you all know anyway. But so we start with a group of cells, and then we get an initiated cell, which is, you see the red dot. That's just a cell with a key mutation, say, for example, a mutation in an oncogene. And then if that oncogene allows the cancer to grow, the cells may grow uncontrollably, and then you have a benign tumor. But at this stage, it's not really a problem to the patient. The immune system of the patient, or for whatever reason, is keeping the cancer in check. It's not growing, but it's not eradicating the cancer either. These two plots represent stage 0 to stage 3 of cancer, which is called carcinoma in situ. But then if you are unlucky enough to pick up an extra key mutation, so the blue, then you may develop a malignant tumor, which is metastatic. And then the cells may detach from the primary tumor and metastasize to other organs, and this is stage 4 cancer. So if you are unlucky enough to have stage 4 cancer, unfortunately the cells may metastasize to your liver, lungs, bones, or even the brain. And so, well, before I go further, I should explain why I'm talking about this, why I'm talking about cancer now, as opposed to statistics and all that stuff. Well, for me, bioinformatics, if you want to be a really good bioinformatician, you have to know knowledge. You have to have knowledge of the medical side of what you're analyzing as well. If you want to just be a bioinformatics analyst, so just literally analyzing data, that's fine, but you will never truly understand the data that you're analyzing. But if you have knowledge of, say, cancer, or of other diseases, or of general biology or medicine, then you will understand better the data that you're actually analyzing. So I'll proceed. So in breast cancer, because breast cancer, I did my doctoral degree in breast cancer. So what is a risk factor? Well, a risk factor is any characteristic or behavior that affects the chances of developing cancer. So in breast cancer particularly, known risk factors, obviously gender, being female is the greatest risk factor, but a lot of people don't know that breast cancer can also affect males. 1% of cases of breast cancer actually occurs in males. Age, so 81% of female breast cancers occur in women aged more than 50 years of age. Family history, I'll go over that later, diagram later. And then a personal history, so if you already have a diagnosis of breast cancer, you are more likely to develop breast cancer again. And so family history, so a lot of people will probably have heard of this gene, this is BRCA1 or BRCA1. So if you have an inherited mutation in this in your family, it results in a 65% chance of developing cancer by the age of 70. And also the number of affected relatives. So if you have one relative who has breast cancer, your risk is doubled. And if you go down to 3 affected relatives, your risk is quadrupled. So weight and diet are obviously risk factors. So obesity is a risk factor. Alcohol, and the reason why alcohol is a problem in breast cancer particularly is because alcohol raises levels of estrogen. Genetic factors, obviously carrying germline mutations. And then hormonal factors as well. So early onset puberty, late menopause, and hormone replacement therapy as well. So how does this relate to bioinformatics? So part of my work at the University of Leicester in England was to derive blood biomarkers for breast cancer screening. So my job was to, as bioinformatician, was to develop genetic markers that we could use to diagnose breast cancer early. So before cancer actually appears, before a tumor actually grows. So the samples in this study, we had over 500 breast cancer and control samples, including tumor DNA, matched blood samples, matched normal samples, and healthy controls. And what did we find? Well, we found evidence of breast tumor DNA in the blood samples even years after the women had surgery to remove their tumor. So I'll get back to that later. And then, so this work also resulted in a lot of publications and an award of, well, in reais, 5,1 million reais. So in this study, we took DNA samples from the tumor, and then from the same patient in which the tumor was present, we took a DNA sample from the blood. So in the blood of everyone, there is a type of DNA called cell-free DNA. So if you take a sample of blood, you can extract DNA from that blood sample. And then we compared the blood DNA to the tumor DNA to see if we could see tumor markers in the blood. And so this work was published in Genome Research, and it also made a book chapter. And this publication is actually now listed in the top 5% of all publications, so it's been a really successful study. And so getting back to a bit to bioinformatics, so some of the results from that study, this is a principal components analysis biplot. So principal components analysis is a very common technique in bioinformatics. And what this shows, so the red dots, these are tumor samples. The blue dots are healthy samples. And then these green dots here are blood samples. So there's a DNA from the blood. And you can see how some of the blood samples are very similar to the tumor. So in those people here, these people are showing signs of tumor DNA in their blood. Because when you have cancer, your tumor will release DNA into the bloodstream, and then you can detect that. And so down here in this other plot, this is a chromosome copy number profile. So if we look along horizontally, this is a chromosome. And what we see here in tumor DNA in the bottom, we see this signal here. And then in the blood DNA, we see the exact same signal. So we can see tumor markers in the blood. And now this plot, what this highlights is, this shows a large genomic alteration in cancer. Again, this is from a patient in my study. So if you look at this is a copy number profile again. So this is actually chromosome 8. So this is a healthy chromosome. You can see the profile is quite normal, consistent. But then in the tumor sample in chromosome 8, what's actually happened is this part of the chromosome has been deleted here. So it's gone. Whereas this part of the chromosome has been amplified. So you get this situation in the tumor, and this is called an isochromosome. And we can see this on the copy number profile plot here. So the signal drops below the red line here, where it's raised above the red line here. And this highlights the idea of genomic instability in cancer, and it shows how unstable the cancer genome is. These are just other publications arising from that work. I won't linger on that. So other work as bioinformatician in the realm of breast cancer that I did was in whole genome sequencing. So in whole genome sequencing, the idea is to determine the entire coding sequence of a person's genome, of the DNA. So we did that for, again, healthy samples, tumor samples, and then also a metastatic sample. So for the computer scientists here, you'll understand that this is quite a lot of data. What whole genome sequencing generates is a lot of data. So this was around 6 terabytes of data, which is a lot. And as a bioinformatician, it's difficult to analyze as well because it's hundreds of millions of different variants or mutations that you have to analyze and look through. And from this study, we highlighted a phenomenon called intertumoral heterogeneity and clonality in breast cancer metastasis. So I'll come back to this term later because it's key in in cancer in general. In these samples, we also identified key mutations. So TP53, a lot of you will know, is a key oncogene in breast cancer. Actually, mutations in this gene occur in the majority of cancers. And in this study as well, I also found a key mutation that was unique to the metastatic sample, which would affect how meiosis and mitosis are conducted. And this mutation would result in genomic instability. I'll get back to this later. That just, again, shows that that data was published. And so back briefly to, again, in the realm of breast cancer, This relates to my work in Albert Einstein College of Medicine in New York, where I was looking at the BRCA1 gene. And I was working on a data set called Phantom 5, which is produced in Riken in Japan. And Phantom 5 is just a gene expression data set. And my role as such was to figure out why BRCA1 is mostly associated with breast cancer, because we know that BRCA1 is associated with breast cancer, but we don't fully understand why it is mostly associated with breast cancer. So BRCA1, this gene, it's just a regular gene on chromosome 17. This is its protein sequence, and there's an actual crystallographic image of the protein itself. Well, what does BRCA1 actually do? So when you have a double strand break in the DNA, so when the DNA actually breaks, when the actual DNA molecule breaks in two, BRCA1 is recruited to the site of damage, and then it recruits the homologous chromosome in order to repair that breakage. But so if you do not have a functioning BRCA1 gene, or protein, what actually happens? Well, no one actually really knows, because no one has studied this. So what do you think? My hypothesis is that this will result in genomic instability, and that's what I showed. And so again, this is a principal components analysis plot, which highlights this notion of genomic instability. So in women who have a mutation in this gene, these women, even if they do not have a diagnosis of breast cancer, they will show signs of, they will have a genomic profile that's similar to cancer. So if you look over here, these are all healthy samples. If you look over on the left, these are all cancer samples. And then if you look down here, these are two women who had a mutation in BRCA1, but they did not have a diagnosis of cancer. So they did not have a diagnosis of cancer, but the fact that they have a mutation in this gene, it still shows that they're actually grouping with the cancer sample. So their blood DNA profile is similar to that of cancer already. So I said I would get back to tumor heterogeneity. So what is tumor heterogeneity? So it's a big research focus at the moment in cancer research. So in a single tumor, there can exist many distinct tumor cell types, and this is called heterogeneity. If you use a drug to tackle just one tumor cell type, you actually permit other cell types to grow more. And I'll show this graphically later. And this results in drug resistance. And actually, when you do this, you can end up in a long losing battle of drugs and treatments. And I show this here. So in any tumor, say, for example, a breast tumor, you can have multiple populations of cells, and each is actually like its own tumor type. So in a single tumor, there can be actually millions of different tumor types. So how do you target each of those? Well, from the tumor, you may develop a metastatic clone again, and you can apply a drug to that, which will kill off that clone, but then another one grows after that. And then you can apply more treatment and to kill that, but then another mutation may develop and another metastatic clone may develop. And this type of cancer is a very aggressive type of cancer, we say. It's very aggressive, so no matter which treatment you apply to it, it will continue to find a way to overcome that treatment. And this is actually kind of like evolution in fast forward. So the cancer is like, the cancer is trying its best to survive, whereas you're trying to kill it off, but the cancer is surviving. And this is a very aggressive type of cancer. It's not in every case, though. And so I say this here. So genomic instability drives or increases tumor heterogeneity. So if you have an unstable genome, you will get into a situation like this where your cancer is very diverse. And this makes a cancer then more difficult to treat. So that's pretty much all my work in breast cancer. I'll now go over clinical diagnostics and genetic testing. So just to explain what clinical diagnostics is. So clinical diagnostics is where, for example, if you have symptoms and you do not know what your illness is, you may go to your doctor, and then your doctor may refer you to a genetic testing laboratory for a genetic test. And there they will test your DNA to see if you have any key mutations in genes. And using information from your DNA, they may be able to diagnose you with an illness. So that's what clinical diagnostics is about. But so why do we actually need bioinformatics and new technology in clinical diagnostics? Well, many hospital laboratories all across the world. It's not unique to Brazil. It's happening in United States of America. It's happening in England. It's happening in Ireland as well. Many hospital laboratories use old equipment that has low sensitivity and specificity. With bioinformatics and new technology, we can diagnose diseases earlier through population screening, for example. And this relates to my work on breast cancer. The data that I produced could be used as a screening program in breast cancer. And we can also permit the stratification of patients into risk groups or permit the tailoring of treatments to patients based on likely response, which saves money. So in clinical diagnostics, there are certain barriers to new technology, which I faced in my career. So the main barriers to new technology in clinical diagnostics, so in hospital diagnostics, well, very pressurized environment. As we know, well, Brazil is in recession now. For hospitals are going to be a very pressurized environment. Everyone is busy, they have too much to do. So it's a highly pressurized environment. And it's the same in the National Health Service in England. I found it's they don't have enough money basically to do everything that they want to do. And then also another barrier is negative or all clear result reporting. So what this means is if you go to your doctor again and then you receive your result back to you, but there's no mutation detected, can we actually trust that result, that there is no mutation detected? Can we trust the technology, that the technology really found no mutation? So that's important, because we do not want to say a patient is healthy if they really are not healthy. The technology must be highly sensitive, of course. Another key problem in clinical diagnostics is gaining trust from clinical scientists and doctors. So I found this as a major barrier as a bioinformatician in the National Health Service in England. I found that actually making the clinical scientists believe me was very, very difficult, because they did not understand bioinformatics. They did not understand new technology. They were stuck with old methods. So I found that a big barrier. And then obviously I've already mentioned tight budgets, so lack of investment. And then because we're talking about clinical diagnostics here, I need to make a distinction as well. So in research, progress is achieved through experimentation, trial and error, as we know. We have the freedom. We need the freedom to make mistakes in order to learn from the mistakes. Whereas in clinical diagnostics, we are closer to the patients and there's a certain fear of making mistakes. So it makes the work more pressurized and more stressful. So, like I said, my job as bioinformatician in the National Health Service in England, it was actually Sheffield Children's Hospital, so a children's hospital is really great to work there. My job as bioinformatician was to replace Sanger sequencing with next generation sequencing in the hospital clinic. So Sanger sequencing, as some of you may know, is the current gold standard in clinical diagnostics. So what I did was I developed a bioinformatic analysis pipeline that has 100% agreement with Sanger sequencing. And this is actually the only pipeline in existence to have reported a consistent and complete agreement with Sanger sequencing. So it's fully capable of replacing Sanger sequencing. And they are still using it in this hospital in England, in Sheffield. And as a bioinformatician in the clinical setting, you have to think of other things, such as guidelines. The methods that you develop have to adhere to guidelines. And also data protection law and patient privacy law, which is important. A lot of bioinformaticians in academia will not think of these things because they work in academia. They don't need to think of them. And that just shows that it, again, was published. Now, this is the actual... I'm not going to explain this in depth, don't worry. This just shows the analysis pipeline that I actually developed bioinformatically. So each step, it processes the data, and then so it takes the raw data at the beginning, it processes it, and here it produces QC reports. And then it keeps going down and down and down and down. And then it eventually produces reports for the clinical scientists, which is essentially a list of mutations in the genes they were interested in. But so this is quite a long process, and it is indeed quite a big program, but all it does is it processes the data. It takes the raw data, processes it in numerous ways, and then produces reports. And it's fully automated as well. I won't linger on this too long. This just shows the different types of data that are produced from that pipeline. So a lot of different types of formats are produced. So again, because this is the clinical diagnostics, we need to focus on privacy. So in clinical diagnostics, I don't know the regulations in Brazil exactly, but in England, we cannot share any patient identifiable data without the prior consent of the patient. And also, we cannot look at mutations in DNA regions not specified by the referring clinician. So if the clinician is only interested in gene X, but we sequence gene X and gene Y, we are by law not allowed to look at the mutations deriving from gene Y because the clinician did not ask for it. So questions to ponder in this regard: who actually owns your DNA code? So I guess I own my own DNA code, you own your DNA code, and all of you own your own DNA code. So your DNA code, the actual sequence, is your private information. It's like your credit card number, your address, your date of birth. It's private information, and it's important to realize that. So don't give your DNA code away to people too easily. It's worth money to people. And then in relation to this point, that we cannot look at certain regions of DNA not referred by the clinician, should an incidental finding be reported if it is a key mutation? So if we actually take a look at a region of DNA we're not supposed to, and we see a key mutation in, for example, an oncogene, should we actually go through the court system and go through the law system and tell that to the patient? And actually, this did occur when I was working in the hospital in England. I actually, as the bioinformatician, I mistakenly took a look at a gene I was not supposed to. And we found a key mutation in a gene related to breast cancer in a woman. And so the director of the laboratory actually went to the court in the city where this was, Sheffield, to gain permission to tell the patient about the mutation. So now I don't know what's happening with that patient, but she will no doubt have to change her lifestyle if she wants to avoid breast cancer in the future. I won't linger on this too long. This is just another work that I was doing in the clinical environment. It's just about setting up, developing a new program to automatically distribute clinical reports from one hospital to another. And this is more a computer science exercise. So again, it's using different types of computer languages, so JScript.net. And it involved editing star limbs. So LIMS is a lot of you will probably have heard of LIMS. LIMS is the Laboratory Information Management System. It's used a lot in clinical environments. And this just gives an example of the code that I developed to do that. So again, it's not, even for me, this is quite complex to do, so I won't linger on that too long. So we're actually near the end of this section. So another practical example of bioinformatics that I was doing was on B-cell lymphoma, which is also a type of cancer, I know. And this was at Imperial College London. So my job as bioinformatician, the group's work was focusing on DNA stress and damage after oncogenic activation in B-cell lymphoma. So if you look at MYC, MYC, and P210, these are oncogenes in cancer. They are very well known in cancer. So we were activating these oncogenes in a B-cell lymphoma setting, and then we were checking to see what happens to the DNA after these oncogenes were activated. And this was an experiment using a technology called chromosome immunoprecipitation. So chromosome immunoprecipitation will return regions of DNA where a protein of interest is bound. So the protein binds to the DNA, and then the instrument will return those DNA sequences where the protein is bound. And the protein of interest that we were looking at was gamma H2AX, which binds to regions of DNA where damage has occurred. So as a bioinformatician, I developed, again, analysis pipelines for identifying these regions of DNA where this protein had bound. And the results showed that after activation of these oncogenes, it results in widespread DNA damage. So this image is like a barcode. So this is just a full chromosome from top to bottom. And this is after oncogenic activation with MYC. This is after oncogenic activation with P210. And you can see that each black kind of scar as such is where damage has occurred on the DNA. And these plots relate to both of these here. And again, this is just a kind of a screenshot, a capture of the tail from one gene. So it just shows a signal of damage over this particular gene. So the damage is quite extensive. So again, this just goes back to the idea of genomic instability. When you activate an oncogene, your genome becomes more unstable because DNA damage is more widespread. Now finally, I could not give a presentation in Porto Alegre without talking about my current work in London in rheumatoid arthritis. So this is what I'm doing at the moment. So part of the work of the department where I'm working in Queen Mary University of London is about defining a gene expression signature of rheumatoid arthritis. So I can't say too much about this, but it's about observing gene expression in rheumatoid arthritis biopsies And our ultimate aim is to molecularly classify different arthritis pathotypes. So research in arthritis is very behind cancer. Cancer receives a lot of money for research, and it has done that for decades. So research in arthritis is still lagging behind a bit. And I don't know if you know about arthritis, but it's about inflammation of the joint. So arthritis is an autoimmune disease, and it results in inflammation of the joints here. So this is just a elbow joint. And in arthritis, currently there are 3 histopathotypes: follicular, diffuse, post-immune. And this is from a publication by my current supervisor, Professor Pizzalis. So my job as bioinformatician is to identify gene expression signatures of each of these. So we're at the final section now. Any questions from that second part? No? Preguntas? No? No? Eu vou tomar água. No mais perguntas? No? Okay. So the final section, it's not very long. The final section is just about the complexity of life and the frontier of bioinformatics. So I guess it's complex, but it's interesting. So life is definitely more complex than we had previously thought, I think. So we have sequenced the entire healthy human genome, but actually we have many unanswered questions remain in genomics. So everyone usually thinks of DNA as just a straight line like this, it's a sequence of letters, but DNA, it's not sufficient to think of DNA in that way, in a linear way, and it's neither sufficient to think of DNA in a gene-centric fashion. So everyone just thinks of genes. Genes are the most important things we should be looking at. That's definitely not true. There's a lot more to DNA than just genes. So we must also consider the 3-dimensional structure and the folding of the DNA molecule, and I show this on the next slide. We have to think about DNA-protein interactions, so where a protein is bound to the DNA. And then finally, a really interesting area at the moment is looking at non-coding intergenic regions or genomic waste grounds. So previously, everything that was not a gene, so outside of coding regions, we thought it was just, well, like waste ground, Lishu, basically. So it's actually been shown that these regions are quite important. So if you look at this image, this is a strand of DNA here, like that. And then, so this is the start of a gene, the 3' UTR. You go along here, this is an exon in the gene. And this is the, so that's the 5' UTR. Here's the 3' UTR. So that's the start of gene, exon, and then the 3' UTR. But what you can see here is that this region of DNA, which we previously thought as waste ground, as functionless, a protein is actually binding to this region of DNA, and then through another protein, it binds to an RNA polymerase. So RNA polymerase is a very important protein in genomics because RNA polymerase will initiate transcription of this gene. So you can see how this intergenic region, this waste ground, is actually enhancing the transcription of this gene. So that's really important. This could be millions of bases away, but it's actually having a functional effect on this gene. So we have to think about that 3-dimensional structure of the DNA. And you get pockets of this along the genome in each cell. So you get highly heavily folded regions of DNA with numerous proteins interacting. And it is in these regions where transcription is occurring. So actually only a very small fraction of our genome, only 2%, relates to genes. So only 2% is transcribed into RNA and then translated into a protein. But actually, up to 70% of DNA is transcribed, but then it's not translated into a protein. So these are non-coding RNAs. So transcription occurs, but then they're not translated into proteins. And these are heavily abundant in the genome. And for most of these, unfortunately, there are hundreds of thousands of them. The function is actually unknown, but for the ones we do know, they do have functions. Some of them are very important functions. So who remembers this? This is the central dogma of life. It's actually no more. Life is not that simple. So we previously thought DNA goes to RNA, and then RNA goes to protein. But we have to cross that out, I'm afraid, because now we get the situation where RNA can bind back to DNA and have an effect on transcription. And we also get a situation where proteins bind to DNA. In the past, when in research, we could not see this happening because we did not have the technology to probe DNA at this level. But we can now see that it's very fluid, where RNA can bind back to DNA and protein can bind back to DNA. So why is all of this a problem? Why is the complexity a problem? Well, due to life's complexity, disease is also complex, unfortunately. So the vast majority of research labs only focus on one part of the problem. And so obviously another problem in research is competition for funding, competition for publications, and personal differences even. These mean that groups don't work together, even in the same university. So in a single group, if we just observe one part of the problem, the only thing that we achieve is to generate more hypotheses and more questions. So we've all seen publications in the discussion and the conclusion. They say, this could do this may do that. It's all conditional tense. No one actually comes up with a concrete idea about what's happening in a disease. It's all the conditional tense. We're near the end, don't we? So, like I said, a lot of research labs are only focusing on one part of the problem. And by that I mean, so if you're researching cancer, for example, you may only be looking at DNA. And so in DNA, if you do whole genome sequencing, like I've done in my career, you can never explain too much. So you can never explain what are intergenic variants doing. You can detect variants or mutations, but you can never explain what effect they're having. You can never explain what enhancer or promoter region variants are doing. You can never explain what intronic splicing region variants are doing. And I say here you can use in silico tools. So in silico tools are bioinformatic tools that attempt to explain what each variants are doing. But I never use these. For me, a bioinformatic tool can never fully explain what's happening in the cell because it's just far too complex. So actually, I never use these tools, full stop. And then so other labs might be doing an RNA sequencing experiment. So when you determine RNA, you're determining gene expression, you're not looking at DNA. But if you see increased transcription of a gene in a disease, well, that's fine, you can see that, but what's the actual cause of that increased transcription? In some diseases, there are actually novel transcripts, RNA transcripts that you may find. But what is the cause of those? And then ChIP-seq, so ChIP-seq is chromosome immune precipitation. It again looks at regions of DNA where a protein has bound. So what's the cause of increased binding of a transcription factor to a particular locus? We don't know what's going on there. And again, a novel disease-specific transcription factor binding site. So the idea is that one group may be looking at this, another group may be looking at this, another group may be looking at this, and they're not working together to actually help each other explain what each disease is doing because they're in competition with each other. So how can bioinformatics help this? Well, bioinformatic methods can help to merge all of those data types together, all those types of biomedical data in a way that is translational. And translation is another key word that's come around in recent years. So translational is about bringing all the data and research into a clinical environment to ensure that that data does not go to waste. And to do this, obviously I go back to this core, bioinformatic core. So to do this, groups could work together through bioinformatic cores. So if you have a bioinformatic core in a university, groups can work through that and work together through that. So bioinformatic core is a centralized bioinformatics facility. I'm not going to explain this in too much detail, but I've been in bioinformatics for 7 years now, and I've obviously got my own ideas. So this is just some of my own ideas. So if you have each of these types of experiments again, so DNA, RNA, ChIP-seq, and then mass spectrometry as well, you can develop novel methods for those. And to ensure that they're highly sensitive and specific, you have to obviously compare it to the gold standard. So I've already done that for DNA sequencing in the clinical diagnostic environment because I've already developed an analysis pipeline that agrees with the current gold standard of Sanger sequencing. And then each of these methods produce different types of data. And then the key point is when you have to develop an integrated bioinformatic algorithm to actually merge all of this data together. And that's not easy. No one actually knows exactly how to do this at the moment. I've talked to a guy last week. He's a top professor in bioinformatics, and he doesn't even have a concrete idea about how to do this. So once you merge all of that data together, then you can bring in clinical and demographic data, so like electronic health records. For example, in a cardiology setting, systolic or diastolic blood pressure, cardiac index, or age, sex, and what does that say? Dietary habits, thanks. And then once you have your bioinformatics data from technology and you have your clinical data and merged together, you then enter a model identification stage. And this is more in the realm of biostatistics now. So you can, for example, apply a logistic regression model. And then you keep rerunning the model essentially until you find the best model that can describe your data. And then once you find your best model or models, you then do a technique called cross-validation, which kind of just checks that your statistical model is sound. And then that's the end. So this is all hypothetical at the moment. So we're on the last few slides. So key points. This is just from the first section again, just to recap. So again, so bioinformatics is the bridge between information technology and biology. Bioinformatics is very broad, as you can see. I've only touched on a small part of bioinformatics here. And a really key point is you do not need to study a course in bioinformatics in order to be a bioinformatician. Obviously, research bioinformatics is very different from clinical bioinformatics. Like I said earlier, there's currently too much data to analyze and too few bioinformaticians. We actually need more bioinformaticians to help us analyze this data. And then obviously bioinformatics will continue to grow further in the future. And just the key points from the last two sections. So as you can see, bioinformatics is not just about analyzing data. You do find a lot of bioinformaticians whose sole job is to analyze data. They give the results back to the clinician and then they have no further say in what happens. Whereas people like me, I have a background in biology, I can actually understand the data that I'm analyzing, so I can give really positive feedback to the clinicians, and I can really actually help to tailor in which route a project will go. So as you can see, as a bioinformatician, you have the opportunity to work on many diseases, and you can be a key player in a group. Bioinformatics can definitely help to make medicine more precise. So in a lot of cases, doctors are applying drugs to people and they don't even know if the drug will work. So bioinformatics can help in that regard. And as you can see, bioinformatics can help to replace outdated methods and outdated technology in clinical environments. And again, I've done that in Sheffield Children's Hospital in England. So just acknowledgements now. So I'm gonna go through each one. So from America, my friend Christoph in New York, and also Mark. I worked with Mark in Florida. From Italy, Professor Pivandi in Milan, and Marcin. From PUCRS here, Professor Teixeira, then Larissa, Renata, and Alice. From Ireland, Paul Kelly from computer science, Tomei from when I was studying biology. And then from the UK, obviously, a large part of my career is from the UK. So Jackie Shaw, my supervisor in Leicester during breast cancer. Charles and Justin, obviously, these are world-renowned breast cancer clinicians. Nicholas, Anne, Darren, Karen, Dave, Mike, who I work with at the moment, my current supervisor, Costantino Pizzalis, a great, kind gentleman. And then from York, that's York in England, Caroline Descent. So, obrigado pelo seu tempo.
Perguntas, comentários?
Yeah, it's rare to find bioinformaticians doing that, but when I started my doctoral degree, for example, I started my doctoral degree in the laboratory. I was doing, pipetting, extracting DNA, and then I was analyzing that data myself. But I think there's just not enough time to work in the laboratory and then work as a bioinformatician as well. You have to do one or the other, I guess, because you in certain situations you find a principal investigator. So a principal investigator leads a research group. When they start a new group, they may not have any staff as such that they may be doing everything. They may be doing the wet laboratory work, the bioinformatic work, and then interpreting the results. But in like more mature research groups, there's like a person to do this, a person to do that, another person to do that. It's more defined. No mice?
I think that they are a very important. There are some very important ideas for us that are the ideas for the future. And there is an urgent need to address these because in some places these are not anymore in the future. Ideas being developed now or have been developed and are being improved. So all the idea of connections, the study of connectedness, and the approach to the complexity of phenomena is, I think this is amazing. Yeah. And that's what you face in your work.
Yeah, well, just on the first point, so all of this is, bioinformatics is relatively new, it's modern. So a lot of the methods that we use, actually, there are a lot of errors in the programs that we use. So typically a bioinformatician will develop a new program, they publish that program, and then they release it to the world to use. But often you find that these programs which are published, they have a lot of errors in them. So as a bioinformatician, you have to kind of double check, treble check, quadruple check what you're doing because there are a lot of errors. So everything is new as such. So it's not like some parts of medicine will be very mature. So like a doctor will know exactly what a diagnosis is just based looking at the symptoms. So everything has been around for a long time. But in bioinformatics, it's only been around for a very short time, relatively speaking. So everything is still heavily prone to error. Everything is still in development. And yeah, the complexity issue, well, it's really difficult to actually, we still don't fully know what is happening inside a single cell, a human cell. It's still too difficult to actually fully explain what's happening inside the nucleus, for example. And so it's just really complex. But as we develop more and more data, I think we'll get ever closer to fully understanding, well, life, I guess. It would be bad to actually predict life, because then we would have no fun in what we're doing. We could say I could predict what I'm going to do in 1 year's time by looking at my DNA or whatever, but that would make life very boring. So, yeah.
There is, you know, that Epicurus, that was a philosopher 2,400 years ago. He was very worried because the vision of the world at that time was, well, they were the atomists. Huh? They were proposing that the ultimate reality are the atoms from Greek, meaning that small bit that you cannot divide anymore. But but their idea was that the atoms were, well, what's his name? Huh? No, professor. You are a physicist, help me please. They thought that atoms had a linear and parallel route, were moving. But then it was not possible to explain the free thinking, because everything would be determined. And you would not have new things. So this is the
yeah, some people don't like the idea that you can predict life, because then it's very boring. Some people think I study a lot of quantum chemistry and quantum physics and all this. So some people will cite the uncertainty principle, Heisenberg's uncertainty principle, to say that we can never fully predict life because the randomness that exists at the very fundamental level. But on atoms, I did work in quantum chemistry once, a very short role, and actually we can only predict the exact state of the hydrogen atom. So the hydrogen atom is the most basic atom on the periodic table. Once we go up to number 2, which is helium, I think, we cannot actually predict the position of the electrons around the helium atom because it's already too complex. With hydrogen, it's only 1 or 2 electrons, so we can predict that. But then the mathematics, when you scale up to just helium, it's far too complex. Yeah, life is complex. So.
Any comments? I have a lot of comments. Algum comentário? Pode fazer em português, a gente traduz.
Português. O que você acha que... What do you think about NI8?
Eu entendo português.
humanos. A China quer sequenciar nos próximos anos 100 milhões, parece, de genomas, e os Estados Unidos também. Tem 100 milhões de genomas humanos.
China.
Então, China. E os Estados Unidos parece que é 100 mil genomas. O que você acha do futuro da bioinformática, essa quantidade de dado que vai ser jogada? Como eles vão ter mão de obra para isso tudo, para lidar com essa quantidade de dados?
Yeah, é muito difícil. Posso falar inglês? So yeah, in England, for example, they have the 100,000 Genomes Project. That's actually located in Queen Mary University, where I am. And so people develop these projects. The government generates these projects because they want to be world leader. Like Estados Unidos always wants to be the world leader, China as well, and Gran Bretaña. But they don't actually think about the bioinformatics, the data side. They just want to sequence the genomes and then forget about it. But then they don't think about the difficulties we will face in analyzing that data. It is difficult. It's a huge amount of data. So like I said previously in breast cancer, I had whole genome sequencing data from, it was just 6 whole genomes. But that was 6 terabytes of data already. So if you have 100,000 genomes, that's going to be into petabytes. So are you a computer scientist or no? Informatico, was it? No. Yes. Oh, so like 1 terabyte is a lot of data. 1 petabyte is an extremely large amount of data. So people devise these projects and they don't actually think about how they're going to analyze it or how they're going to use the data. So, and that's actually a major problem. I'm very close to that project in England. It's in Queen Mary University. And again, one of my bosses is the director of bioinformatics at the university. He's heavily involved in that project, and he is very angry. Ele fico bravo com tudo isso, porque as pessoas não pensam na bioinformática. Ok, elas vão fazer com os dados enormes.
Can you talk a little bit about how is your daily work remotely in Porto Alegre, facing London and US?
So yeah, well now it only takes me 5 seconds to walk to work because I get out of bed and then I walk to my desk, which is 5 seconds away. So it's the shortest commute that I've ever had. It's a bit different, but you obviously need to be very concentrated and focused on what you're doing. And how I do it is, so I have my laptop and then I have another screen. So I log into my computer in London through remote desktop connection. And so it's like a virtual desktop as such. And it's actually easy. I have teleconferences almost every day with people back in London. And because I'm a bioinformatician, a lot of time I can go for 1 week, 2 weeks, just analyzing data. And then I arrange a teleconference to report the results back. So like some of my colleagues in London, they are clinicians and they have to see patients every day. They have to go to the hospital to work. Whereas I do not have to. I can, I mean, one of the best examples is I was returning from London one day, London via Sao Paulo. And while I was in Sao Paulo airport, I logged into my computer in London. I was waiting in Sao Paulo in Guarulhos. So I logged into my computer to do some work in the airport. So I can do my work from anywhere in the world, basically. I could be sitting on the Copacabana doing my job, but obviously I'm here in Porto Alegre doing my job. So it's a different situation, but it's good to be able to work from home.
Any other question, comment?
What do you think about entrepreneurship and bioinformatics in Brazil?
Which was the first term you used?
Entrepreneurship. Empreendedorismo.
Startups. And in Brazil?
Yes.
Well, you mean, like, for example, you are thinking of starting a new company in Brazil, or what? or just the state of bioinformatics in Brazil, or in general? Well, I mean, in Brazil so far, bioinformatics is not as extensive as in Estados Unidos or Gran Bretaña. I think one of the only other groups that I know of is in Sao Paulo, in Campinas, University Campinas. There's a guy there who actually, I forget his name, but he's a bioinformatician. He's leading a new bioinformatics group there. But other than that, bioinformatics is not really big in Brazil. I've not seen much happening. So I guess for a person like me, it's always better to to continue working with Queen Mary University in London, because they will be producing more data. And the problem with Brazil as well, it's a beautiful country, but it's in recession at the moment. So and unfortunately, a lot of money is going to be cut for pesquisa research. So I don't know what that will mean for like bringing new technology to academic environments or hospital environments. So it looks like for the next few years at least, we will be using well, I don't expect bioinformatics to increase that much in Brazil in the next few years until everything improves. But Brazil definitely has the potential to grow a lot, I think. It's a big country. If it can get things right at the top, it can definitely grow a lot, and its economy can definitely grow big.
Yeah, I believe that in a crisis moment, you have a lot of opportunities. And I think that we go back to that idea that we should change our mind and attitudes. And we need that with the people that have money, those that have money to invest. And in Brazil, we have a lot of problem with this, because people that have money don't want to face risks. And facing risk is important. This is the difference. But I think that crowdfunding, you know better than I, is a very nice alternative nowadays, and it connects you with all the world. So you may find investors in Singapore or in England or
China.
Anywhere.
China.
China, that's a good place to find investors. So I think that
The same thing happened in Ireland. I'm from Ireland. The economy crashed in 2008. And now agora, ainda para mim, now I'm trabalho na Irlanda para Bioinformática, because now the economy of Ireland is only beginning to improve again now. It's 2016 now. The economy crashed in 2008, so nearly 10 years after the economy crashed, it's beginning to improve.
it's very interesting to notice the age of the audience. Have you noticed? I noticed, because I am in every forum for 6 years. And we have a lot of young people, including Walter over there. Very young people. I think this is very interesting, because the title Bioinformatics probably has kept the attention of young people. This is the future. So on Monday, we received a group from Cambridge University here. And they came to talk and to discuss about a proposal of a project. Well, they are running already a project that is, the acronym is BRIDGE. It's very long, but it's about climate change. And there were 3 professors and 1 PhD student. All of them, of course, one is a PhD student, but the other 3 professors are very young, very young. Kevin is very young. You were very young. So this is a sign. And from Cambridge, 3 very young professors at Cambridge. And one is professor both at Cambridge and at a university. The name is very difficult, from Netherlands. So it has a
Nijmegen or Groningen. The Netherlands has a lot of strange names.
Yes, strange names. And they are studying climate change. And one of the professors actually linked it to the Open University in London. and she's a geographer, and she's Chinese, and she does not speak English very well, you know, but she's working in Open University in London. And two other professors are French, Jean-François, I don't remember the name of the other. And the PhD student is from Chile. I'm writing a text about this, and you are knowing this text in advance, premiere, because it's very, very interesting. This group came from Cambridge University in order to try to establish cooperation here, and they are very young. They all are very young. One is a geographer, but works not only with geography, works with legislation, economy, I don't know what else. One of the French professors, he is originally, he is a physicist and has been working with subatomic particles. And now he is working with climate change and is working with sociology of climate change and economics of climate change. And he talked that he's studying economics especially because he wants to find good reasons for people that have money to face the proposals to reduce for example, carbon dioxide emissions. Trying to produce scenarios where the reduction of carbon dioxide emissions will be more profitable than the opposite. This was very interesting because they had different formations, and they were not talking about we are doing interdisciplinary research. They are just doing. What is the future? That we may have our track, we must have our track, but you must have an open mind so that you can talk to other specialists and cooperate with them.
Yeah. Well, the University of Cambridge, they cover everything, and they're number 2 or 3 in the world. They're a great reputation. They cover all sorts of topics, so it's no surprise that they'll be aiming to lead on climate change as well, because they try to lead on everything as being one of the top universities in the world. So.
Well, I was, so the internationalization, what more can you expect? 3 professors from Cambridge University. One is Chinese, the 2 other are French. And the PhD student was a Chilean. No one was British. But this you see in Harvard University. Harvard University has a defined policy to promote internationalization. They want people from other cultures, other countries. We have Kevin here today. This is very nice for us because we are hearing his personal experience. He came from Ireland, and he has been working in England, and he has been working with several other groups from several other countries. So this is very important. So you, the young people here, this is the way ahead. Globalization, interdisciplinarity, and facing the complexity, facing the challenges. That's a thing that most of the old professors, they cannot do anymore. Some of them perhaps will change, but not all of them. So this is the future ahead, I think. I don't know, what's your idea with your experience in graduation?
So if, for example, when I studied computer science originally, if I stopped then and did nothing after computer science, I would probably be still in Ireland working in computer programming. But then I decided to do biology. I completely switched. And doing biology opened up the door to doing a PhD in breast cancer in England. And then that opened up the door to everything else. And I guess one of the key moments was when I decided to call myself a bioinformatician myself in order to bring back the computer science experience that I had. So when I started calling myself a bioinformatician, Kevin the bioinformatician, everyone started listening to me more because there is a great need for bioinformatics. So if you're a computer scientist here, or you're an engineer even, or a biologist, you can probably easily transition into bioinformatics, and then the amount of career opportunities opens up even more. And just so historically, doing a medicine degree would open up the door to a lot of travel, and working in a lot of different places. But actually, if you do bioinformatics as well, it opens up a lot of travel opportunities as well, because particularly United States of America and Europe, there's, for example, I worked in Italy as well. There's a group in Italy that they had a need for bioinformatics, so I went there just for experience, for life experience, and it was really good to work in Italy. They have really nice food. So, so I guess what I'm saying is, another key thing about my career, so this is about my personal experience, is you can kind of dictate where you want to go in your career. You do not have to essentially listen to, you can basically try to forge your own career to decide where you want to take it. You do not have to just sit around and wait for an opportunity to rise in something. You can also train yourself in something new, and then you can change your career path. So there's a lot of opportunities. I guess that's what I'm trying to say.
Yes, and we have, oh, sorry.
What advice would you give to someone that is not a computer scientist but is interested in bioinformatics?
What did you... O que você estuda? Biotechnology. Well, that's pretty good. I mean, that's a good start. You could... I presume you studied like some computational... yeah. So you could probably easily change your CV, your resume, give it a more informatics focus, a bioinformatics focus, make your bioinformatics skills reflect more on your resume, and then you could actually change your career path eventually. So like I said, I called myself a bioinformatician one day, but then it still took about 1 year, 2 years to actually fully go down the bioinformatics route. You know, it's not just a simple change. But so biotechnology is a really great start. I'm guessing you could easily change into bioinformatics. And a key point as well, in the wet laboratory, I think jobs in the wet laboratory, they're going to disappear. They're not going to increase, put it that way. A lot of postdoctoral scientists that I know, they find it very difficult to find work at the moment, whereas all the work is now coming into bioinformatics because everyone is generating data. So my advice to you would be to try to think about how you could change into bioinformatics, because it would be very fruitful, and biotechnology is a really great start for that. Also Ireland, actually, biotechnology is really important in Ireland, so if you wanted to go to Ireland, you could easily go there.
So Professor Walter is he's in charge of bioinformatics extension course that we call here. It's not in the formal curriculum, but it's an opportunity for formation. Do you want to say anything about that?
Thank you for the opportunity. You have 2 courses. One is undergrad level, and the other one for specialization. It's going on right now. It's the first group that is taking this course, and you expect to have another one starting in the next year. So you still have to improve in this field, but the opportunities are beginning here.
So you have an undergraduate in bioinformatics?
Yeah.
Like 4 years, I guess, 4 years?
No, it's not undergrad. It's one specific course.
Oh,
it's
okay.
not.
This is
it's only to teach Python.
Oh, okay. Yeah.
Specific skill for this
This is
student interested.
Yeah. This is an area I'm not too big on. So like training in bioinformatics, it's an area that because it's difficult to find time to develop training material for other people. So this is an area I'm not too experienced on, I guess. I am like developing training programs in OR, programming language, for other people at the moment. I've got lecture notes in OR. But yeah, I mean, it's difficult to find time to develop training material for people. But it's good to see that the university is developing courses in bioinformatics. It will certainly be good for Brazil. Because actually, information technology in Brazil is actually a really strong point in Brazil. I think it's one of the most connected nations on earth, actually. Maybe South Korea is the most connected with information technology, but Brazil is also big on information technology. So I guess in that regard, it has a huge potential to develop bioinformatics training. So maybe in the future we will see all of the bioinformaticians will be Brazilian, and so that would be good to see.
And there is, well, the Directory of Research, PUCRS, is working with the bioinformaticians we already have. Some of them are here. And in order to organize a structure that is not, this is not easy because it's not a vertical structure. We at the universities, well, the institutions usually are very vertical and disciplinary organized. So you have biology and you have informatics and you have engineering. And then you have groups that are transversal. And you have bioinformaticians that may be having their main activity in the physics school, but they are linked to those that are working at the biology school. So we are trying to design this transversal organization in an institution that is essentially vertical. So that's I think that's a great challenge, but I think it's nice to face it. Any other question?
So this is kind of a personal question. So in your opinion, what do you think about the trust of research molecular biologists in bioinformaticians? Because I'm studying bioinformatics, and we have how do you say class? A course, yeah, discipline, and we had to do a research, and we went through some labs, and we asked, and I can say that 80% of the researchers, they don't trust other people with their data.
Yeah.
So, in your opinion, do you think it's gonna change sometime, or?
Well, I guess you have to be, as a bioinformatician, you have to be very convincing that what you're returning to the people is correct. I do have the same problem though. If I take someone's data, I can see that they do not want. They generally don't feel comfortable with me taking their data and then analyzing it for a while and then returning it back to them. And I can see their perspective. And this is actually why those people that give us data are actually learning bioinformatics themselves. So doctors, they're learning their own bioinformatics skills so that they can process their own data. So this is actually a big problem I faced in the clinical diagnostic environment in the Children's Hospital in Sheffield in England, where I arrived as the lead bioinformatician, and absolutely no one trusted me. Everyone was against me because they did not want bioinformatics in their laboratory. So it was just me and my supervisor, Darren, who coincidentally comes from a business administration background. And so the way that we gained the trust of these people was actually to just connect to them on a human level. So I'm Irish, I'm good at making jokes. So I connect to people by smiling and just connecting on a human level. And when you connect to them on a human level, they believe you more. And so that's it. So it is difficult, it's a problem.
Very recently I learned that the neurosciences has already enlightened an explanation for this difficulty between groups. And they say that in the evolution of our brains, the categorization is a very ancient ability. That means very easily I identify with my group and I keep away from the others. And this attitude that Kevin has just cited is the opposite. I open myself so that I facilitate the interaction.
Yeah, no, well, I think it's to do with being Irish, because being Irish, we are very open to everyone. We've no problems with anyone on earth. So we're very open to everyone. I can talk to pretty much a computer scientist. I can talk to doctors. I can talk to the president, well, former President Dilma Rousseff, on the same level as I talk to anyone else. I don't see differences in people in authority. I just talk to everyone in the same way. So it's probably more to do with being Irish, the Irish genetics, than anything else. So.
I think it's time we would like to thank Kevin very much for your presence here today and your speech. It is really valuable to all of us. Thank you very much, and I hope that we can keep going our a close contact so that we can improve bioinformatics.
Definitely.
Thank you very much.
Thanks.